So next, the embryonic period, specifically we have mentioned in our preamble, starts from the 4th to the 8th week. Specifically, we talk about the ectoderm derivatives, generation, mesoderm derivatives, the two coilems and forging, development of blood and the blood. Okay, so the 4th week through the 80th week of development are very significant in embryonic development because all major organs appear during this time. So the term organogenesis refers to the formation of body organs and systems. And by the end of the 80th week, all of the major body systems have begun to develop, although their functions for the most part are minimal. Then during the 4th week after fertilization, the embryo undergoes very dramatic changes in shape and size, so nearly tripling its size. Then it is essentially converted from a flat two-dimensional trilaminar embryonic disc to a three-dimensional cylinder, which occurs through the process called embryonic folding. Then the cylinder consists of endoderm in the center, basically the gut, and the ectoderm on the outside, on the epidermis, and the mesoderm in between. Then the main force responsible for embryonic folding is the different rates of growth of various parts of the embryo, especially the rapid longitudinal growth of the nervous system, specifically the neural tube. Then the folding in the median plane produces a head fold and tail folds. Specifically, we call it the folding in the horizontal planes and results in the two lateral folds. Then overall, due to the foldings, the embryo curves into a C shape. So this is what we'll be discussing. So this is more of a summary of what actually will be happening. Okay, so that's a summary. So our first things first, we're going to start with the ectodermal derivatives. Specifically, we are going to start with numeration. So remember last time we ended on the notochord formation, so we are going to continue from there. So the notochord and the precoidal mesoderms induce the ectoderm to thicken and form the neural plate, which is known as the neuroectoderm. So this is in the 3rd week. So like I said, remember we ended on the notochord in the 3rd week, you now continue with the 4th week. So by the end of the 3rd week, the plate invaginates in the midline to form the neural groove with thickened neural folds. The folds begin fusion at the level of the 5th somite, this is in the cervical region, and proceeding both caudally and cranially. Then the cranial neural pole crosses at day 25, this is 18 to 20 somites, and the caudal neural pole crosses at day 27, this is the 25th somite. Okay, then the CNS, thus represented by a closed tube with narrow caudal end, which will be our spinal cord, and the broad cephalor end, which will be our brain vesicles. So what we are just talking about here is that the neural groove will start being formed. Then the neural groove starts being formed. Then we have the two poles. You have the cranial pole and the caudal cranial neural pole and the caudal neural pole. That's what we are talking about. So that's how we are going to even learn in the next slide. Okay, so let's go to the next.
By day 16, the embryo consists of 3 layers: endoderm, mesoderm, and ectoderm. Ectoderm develops into the nervous system, and the first phase of this process is known as neurulation. Around day 18, part of the ectoderm begins to thicken. This portion is known as the neural plate, in dark blue, between the cranial end of the embryo and the primitive node. Neurulation is the process of neural plate becoming neural tube. got a schematic animation here to try and illustrate that process. We'll break this down day by day throughout the rest of the video. On day 19, the neural plate begins to buckle in the midline. We call this dip the neural groove, and the mounds either side the neural folds. These folds progress toward each other and begin to fuse around day 21. Where they have fused, they form a hollow cylinder, which is the neural tube. Fusion then continues in synchrony toward caudal and cranial ends. The as yet unfused portions are known respectively as the cranial neuropore and caudal neuropore. Pore literally means minute opening. The minute opening at the cranial end closes around day 24. And the caudal end closes around day 26. A particular group of cells reside at the peaks of the neural folds. These are the neural crest cells, which form a layer over the top of the closed neural tube. These develop into most of the peripheral nervous system. To sum up, neurulation is the process of neural plate becoming neural tube. The closure of the neural folds begins in the centre and zips up towards either end. The central nervous system is developed from the neural tube. It's worth mentioning that by this gestational age, the embryo has started curving into the fetal position. We've kept the ectoderm flat to keep our schematic version really simple, but these pictures from Gray's Anatomy accurately show the embryo at around 21 and 31 days. The neural tube curves through the back of them, like so. That's it for now.
Okay, so this was what we were talking about. So after the animation, let's now, since we have seen what was happening, let's now look at the theory part. So as the neural tube is formed, we said it detaches from the surface ectoderm and lies deep to it. So during this detachment, some of the neuroectoderm cells at the inner border of the folds undergo epithelial to mesenchymal transition and detach from the neural tube to form a flattened irregular neural crest mass, from which the neural crest mass cells migrate, and the cells can also migrate even before the neural folds fuse. Yeah, so the remaining ectoderm, which will be a surface ectoderm, forms the epidermis of the skin. So this is what happens. Okay. Then the neurocrest cells from the trunk region leave the neurofolds after closure of the tube in this region. Then they migrate through one of two pathways. Can migrate through the dorsal pathway, where they migrate through the epidermis, and the epidermis through pores in the basal lamina of its cells to form the melanocytes of the skin and hair follicles. Then we have the ventral pathway, where they migrate through the anterior half of each somite, which will be the derivative of the paraaxial mesoderm, and form spinosensory ganglia sympathetic and enteric, the GIT, ganglia. Then you have the Schwann cells and the cells of the adrenal medulla, but not necessarily the cortex. Okay, that's the neurocrest cells and how they are going to go about after closure of the neural tube. Okay, they start to migrate in the two pathways, the ventral, the dorsal. Okay, then the neurocrest cells from the cephalic region migrate before closure of the neural folds and contribute to skull bones, cranial ganglia, glial cells, and melanocytes. Okay, that's what they contribute to. Then let's also talk about other ectoderm derivatives. What are the other ectoderm derivatives? So after closure of the neural tube, the bilateral ectodermal thickenings, the otic and the lens placodes, which will be the future vestibulocochlear apparatus, and the lenses, respectively. Okay, so in short, the otic will be the vestibulocochlear, then the lens placodes will be the lens, of course. Then these are going to appear in the cephalic region, and they will both invaginate and form their future derivatives. Then there are also the limb ridges. So these are the limb ridges. So we have seen the limb ridges here. Okay, limb ridges. So the limb ridges that stimulate the one which are going to stimulate development of the upper and the lower limbs. Okay. So we are done with the ectoderm derivatives. Now we are going to talk about the mesoderm derivatives. So the thin sheet of the intraembryonic mesoderm differentiates into three parts around the middle of the third week. So you have the paraaxial mesoderm, which is thickened. You have the intermediate mesoderm, which is relatively thin, and you have the lateral mesoderm. You have the lateral plate mesoderm. This is where the small space appear in the lateral place and join to form a cavity, which is the intraembryonic coelom, and thus dividing the lateral plate into somatic, to apparato, and the spratchnic or the visceral layer. Okay, so this is the mesoderm derivative. Specifically, the lateral plate mesoderm is going to divide into the somatic or the apparato or the and the spratchnic, which is our visceral layers. Okay, so this is what we are talking about. Okay, then let's talk about the intraembryonic coelom, which is the body cavity. So the primordium of the body cavity begins as isolated coelomic spaces. Okay, and in the lateral mesoderm and the cardiogenic area, which coalesces to form a single horseshoe-shaped cavity that communicates laterally with the extraembryonic cavity. So we're talking about the formation of the intraembryonic coelom, or body cavity, which is the primary body cavity which begins as an isolated coelom space. So this, of course, you will find that there are horseshoe-shaped cavities going to form, and that horseshoe-shaped cavity communicates laterally with the extraembryonic cavity. So that's what we are saying. So that's where actually even the organs and the organs will start being filled inside. Okay. So the intraembryonic coelom can be understood by imagining a horseshoe of a horseshoe of cavity placed within the mesoderm of the embryo. So this, I'm sure you have seen, looks like an horseshoe. Okay, and they have been pressed inside the mesoderm of the embryo. That's what we are talking about. Okay, so we are done with the derivatives, the mesoderm and ectoderm derivatives. We are now on the folding of the embryo. Okay, so the flat trilaminar embryonic disc folds into a 3D by 4 folds. So what are those 4 folds? Number 1 fold, you have the head or the cephalic fold. Secondly, you have the tail or the caudal fold. And you have 2 lateral folds. And all these are stimulated by the development of the central nervous system. Then the growth of the forebrain beyond the buccopharyngeal membrane results in the head fold, which pushes the heart, the pericardial column, and the septum transversum down and incorporates a portion of the yolk sac, forming the foregut, in addition to placing the buccopharyngeal membrane at the future side of the mouth. So that will be our embryonic folding. That's how it starts to fold. Okay. Then the primordium of the spinal cord stimulates the tail fold, by which it pushes the connecting stalk, the ventral aspect of the embryo. Then it also incorporates a portion of the yolk sac, forming the hindgut, in addition to incorporating part of the allantois to the body of the embryo, and shifting the position of the cloaca membrane to the site of the future anus. Okay, so what we're talking about is this. So if you have seen, you have the allantois, the hindgut, and you have the cloaca membrane here, which will be our future anus. Okay, so what exactly is going to look like? So remember, this was how it was. Then it started to form. You have the 4 folds, and it continued. We have the 2 folds, then it continued and made the lateral folds here. Then finally it formed, and we had the hindgut, the midgut, and foregut, and the cloaca membrane, the allantois, and the cloaca membrane. Okay, and the folding of the embryo is actually cephalocaudal folding. Okay, so we are talking about the head fold and the cephalocaudal or the cephalocaudal folding. So let's talk about the lateral folding. So how is it going to be formed? So the spinal cord and somites stimulate lateral folding, which causes incorporation of another portion of the yolk sac, forming the midgut and the ventrolateral body walls. Okay, the ventrolateral body walls of the embryo are formed. Then connection with the yolk sac is reduced to yolk stalk or the ompharoenteric duct, which is the site of the future umbilicus. So this is what we are talking about. If you have seen the foregut, the midgut, then you have the connecting stalk between the yolk sac and the midgut. Okay, then we are saying the connection with the yolk sac will be reduced to the yolk stalk, which of course will be our ompharoenteric duct, and will be our future site of the future umbilicus. That's what we are talking about. Okay, so like usual, most of the things that require imagination are things I want to use animations, and of course, this was not one of my animations. We use these ones to help you actually visualize what's happening in embryonic folding. Okay, so let's start. And if you haven't subscribe and hit the like button, and also make sure you turn on your notifications to receive notifications whenever you post a new video.
During the 4th week of development, a period of rapid growth, the embryo begins to change shape from a flat trilaminar disc into a cylinder, a process known as embryonic folding. Embryonic folding occurs in 2 planes, the horizontal plane and the median plane, and is the result of differing rates of growth of the embryonic structures. Folding of the embryo in the horizontal plane results in the development of 2 lateral body folds. Folding in the median plane results in the development of the cranial and caudal folds. Folding in both of these planes takes place simultaneously, resulting in the rapid development of the embryo. The cylinder consists of 3 layers derived from the trilaminar embryonic disc. These are the endoderm, the innermost layer, the ectoderm, the outermost layer, and the mesoderm, located in between. The endoderm of the trilaminar disc is mainly responsible for the formation of the gastrointestinal tract. As embryonic folding continues, the endoderm moves towards the midline and fuses, incorporating the dorsal part of the yolk sac to create the primitive gut tube. The primitive gut tube differentiates into 3 main parts: the foregut, midgut, and hindgut. The foregut can be seen at the cranial or head end of the embryo. It is temporarily closed by the oropharyngeal membrane, which at the end of the 4th week of development ruptures to form the mouth. The midgut lies between the fore and hindgut and remains connected to the yolk sac until the 5th week of development. As embryonic folding continues, the connection to the yolk sac narrows into a stalk known as the vitellin duct. The hindgut lies at the caudal or tail end of the embryo. It is temporarily closed by the cloacal membrane, which during the 7th week of development ruptures to form the urogenital and anal openings. As a result of embryonic folding, the major body plan is established, and the 3 germ layers continue to differentiate, giving rise to their own specific tissues and organ systems.
Okay, so that's what we need to understand. So in summary, embryonic folding occurs in 2 planes simultaneously and is a result of differentiating rates of growth of the embryonic structures. They will have the ectoderm, which is primarily responsible for formation of the gastrointestinal tract. Then the gut tube is divided into 3 main parts: the foregut, the midgut, and hindgut. Then the 3 germ cell layers differentiate, giving rise to their own specific tissues and the organ systems. So this was what we are talking about in embryonic folding. Okay, so this was what we are talking about. Okay, so let's continue with our body cavity. So remember we talked about the intraembryonic column, which is the body cavity. Now what is going to happen as a result of the lateral folding? Okay, that's what we are going to discuss. So as a result of the lateral folding of the embryo, the communication of the intraembryonic cavity is narrowed to a very small area around the umbilical cord, which later when the amniotic cavity obliterates most of the extraembryonic cavity, the communication is completely lost. Okay, so this was one of the things we mentioned, and in the lateral folding as the embryo, as a result of the lateral folding of the embryo, the communication in the extraembryonic cavity is actually narrowed. Okay, after it has been narrowed, then you find that it's narrowed to a very small area around the umbilical cord. Then later, when the amniotic cavity obliterates most of the extraembryonic cavity, then that's when the communication will be lost. Okay, so this communication, that's when it will be lost. So let's go to the paraxial mesoderm. Remember we talked about its derivatives. We talked about the paraxial and whatnot. So we are now on the paraxial derivative of the mesoderm. So the paraxial mesoderm is organized, so specifically cephalocaudary, into segments known as the somatomeres and the somites. So the somatomeres are more loosely organized in the head region, forming in association with the segmentation of the neural tube into neuromeres. Then the somites, which are more compact and defined regions, forming from the occipital region, caudary, and first somite forms on the 20th day, and the last pair at the end of the 5th week. Okay, so that's the para. So what are the fate of the somites we are talking about? So each somite can be divided into two main portions. You have the sclerotom and the demomyotom, and it also receives its own segmental nerve component. Okay, so the sclerotom is the ventromedial portion of the somite, which forms a loosely organized tissue, which will be the mesenchyme, and migrates around the notochord and spinal cord, forming the vertebral column. Then, in addition to forming the tendons for its muscles. Okay. Then, what about the demomyotom? How does it go about? So the demomyotom is the dorsolateral portion of the somite, and it can be divided into three parts. You have the ventrolateral lip, which you have a VLL, and you have the dorsomedial lip, which you have DML, of the muscle forming cells, and the remaining dorsal dermatome. So the ventrolateral lip cells migrate forward and form limb and body musculature. Then the dorsomedial lip cells migrate down the aspect of the dermatome and form the muscles of the muscles of the back. Then you have the dermatome, which forms the dermis and the subcutaneous tissue. Then throughout migration, then these cells retain their original segmental nerve component. Okay, so that's what we are talking about, the fate of the somites. So this is the fate of both the somite and the demomyotom. Okay. So we are done with the paraxial mesoderm. Let's now look at the intermediate and the lateral plate mesoderm. So the intermediate mesoderm later forms the urogenital system. Then the lateral mesoderm divides into the somatic and the splanchnic layers. So the somatic layer contributes to the ventral lateral body wall and forms the parietal layer of the body, while the splanchnic layer contributes to the wall of the gut and forms the visceral layer of these membranes. So remember, we talked about the somatic layer, which is the first layer, and remember, somatic most of the time we talk about the ventrolateral body wall. Okay, and it forms the parietal layer of the body. That's somatic. What about the splanchnic? The splanchnic layer contributes to the walls of the gut and forms the visceral, soft internal organ. So the visceral layer of this membrane. Okay, so we have visceral and you have our parietal. Okay, have been formed. Okay, so that brings the end of the mesoderm derivatives, the ectoderm and the mesoderm derivatives. So let's look at the development of the GIT. So as they move toward the midline, the lateral folds incorporates the dorsal part of the yolk sac into the embryo as the primitive gut. Then this will be our forerunner of the gastrointestinal tract. Then the primitive gut differentiates into an anterior foregut, at an intermediate midgut, and the posterior hindgut. The foregut develops into the pharynx, esophagus, stomach, and part of the duodenum. Then the midgut is transformed into the remainder of the duodenum, jejunum, and the ileum, and the portions of the large intestine. Then you have the cecum, appendix, ascending colon, and most of the transverse colon. Then the hindgut develops into the remainder of the large intestine, except for a portion of the anal canal that completely that develops from the proctodeum. Okay. Then separating the cloaca from the proctodeum is the cloaca membrane. So during embryonic development, the cloaca divides into a ventral urogenital sinus and a dorsal and rectal canal. So as a result of the tail folding, the cloaca membrane moves downward and the urogenital sinus, anorectal canal, and the proctodeum move closer to their final positions. So when the cloaca membrane ruptures during the 7th week of the development, the urogenital and the anal openings are then created, and thus the GIT will be formed. Okay, so let's talk about the pharyngeal arcs. So in addition to embryonic forging, development of the somites, and development of the neural tube, we also have 5 pairs of the pharyngeal arcs or the brachial arcs that begins to develop on each side of the future head and neck during the 4th week. So these 5 paired structures begin their development on the 22nd day after fertilization and form swellings on the surface of the embryo. So each pharyngeal arc is or consists of an outer covering of ectoderm and an inner covering of the endoderm. So with mesoderm in between, within each pharyngeal arc, there is an artery, a cranial nerve, a skeletal cartilaginous rods that supports the arc, and a skeletal muscle tissue that attaches to and moves the cartilage rods. Then on the ectoderm surface of the pharyngeal region, each pharyngeal arc is separated by a groove called the pharyngeal cleft. Then the pharyngeal clefts meet corresponding balloon-like outgrowth of the endoderm pharyngeal lining called the pharyngeal pouches. So where the pharyngeal cleft and pouch meet to separate the arcs. Then the outer ectoderm of the cleft contacts the inner endoderm of the pouch, and there is no mesoderm in between. Okay, so we're talking about now what will be the pouches will now start to being formed. Okay, then each pharyngeal arc of the cleft and the pouch will give rise to a specific structure in the head and the neck. So hope you have paid attention. Okay, so pay attention to the pharyngeal cleft and the pharyngeal pouches and what they are going to form. Okay, so this is what we are talking about. So I'm sure you have seen the pharyngeal pouches, which are here, are forming there, and below these pharyngeal arcs, we have the pharyngeal clefts in between. Okay, so that's what we are talking about. So let's move. And remember, in our preamble, we also said each pharyngeal arc contains a cartilage, a nerve, a muscle. That's what we were talking about. So what are the derivatives of the pharyngeal arcs or the pouches? So we have the pharyngeal arc, which is number 1. We have the mandibular, which have the cranial nerve number 5. The muscles it has, we have the muscles of mastication, the masseter, the pitagoid, and etc. etc. Then the skeleton, we have the mandible, maxilla, the malleus, and the incus. Then we also have the hyoid pouch, the hyoid arc, which has the cranial nerve number 7, which is the muscles it has, we have the muscles of facial expression of the epicuraris oris, the pragmas, and the skeleton it has, we have the parts of the hyoid bone. Then we have the pharyngeal arc that contains the cranial nerve number 9, which has muscles the stylopharyngeus, and the skeleton we have parts of the hyoid bone. Then you also have the pharyngeal arc 4 to 6, we have the cranial nerve number 10. And the muscles it has of the constrictors of the pharynx. Then the skeleton we have the laryngeal cartilages, the thyroid, cricothyroid. We have the arytenoid and the corniculate. So the endoderm of the pharyngeal pouches actually gives rise to a number of endocrine glands and part of the middle ear. Then in subsequent order, the pouches give rise to a number 1, we have the middle ear cavity and the auditory tube, which is beer pouch 1. Then the second one, we have the stroma of the palatine tonsils, they are made by the pouch 2. Then thirdly, we have the inferior parathyroid gland and the thymus, which are made by the pouch 3. Then the superior parathyroid gland and the brachial body are made by pouches 4 and 5. So the pharyngeal cleft gives rise to only one structure, which will be the external auditory meatus. So we have the pharyngeal cleft and the pharyngeal pouches. We have done the other. So let's look at the formation of the ear, eye, and the limbs. So the first sign of a developing ear is a thickened area of ectoderm. We have the otic placode, or the future inner ear, which can be distinguished about 22 days after fertilization. Then a thickened area of the ectoderm called the lens placode, which will become the eye, also appears at this time. Then by the middle of the 4th week, the upper limbs begin their development, outgrowth of the mesoderm covered by ectoderm called the upper limb buds. But the end of the 4th week, by the end of the 4th week, the lower limbs buds develop. The lower limb bud develops. So meaning by the end of the 4th week, the lower limb buds are going to develop. Okay, and also the upper limbs that have developed. So next is the art. The art also forms a distinct projection on the ventral surface of the embryo called the heart prominence. Then at the end of the 4th week, the embryo also has a distinctive tail, and you can even actually identify this to say, okay, that's a very distinctive structure. So now let's talk about the formation of the blood and blood vessels. So blood vessels are formed in two ways. We have the first way, which is the vesiculogenesis. So vesiculogenesis is the process where blood vessels form from blood islands. Then we also have the other way, which is the angiogenesis. Angiogenesis is the process where new vessels are formed from existing ones. So blood islands are composed of specialized mesenchymal cells called the hemangioblasts. So these cells are derived from mesoderm. Then such mesoderm cells are induced to become hemangioblasts under the effect of the structure VEGF, the vesicular growth endothelium, okay, factor. Then these are the VGF is actually released by the neighboring mesoderm cells. Then the cavities are going to appear within the blood island. Then you have the central cells, which become hematopoietic stem cells. Then you have the ancestor of all types of blood cells, while the peripheral cells become angioblasts for the formation of the vesicular endothelium. Okay, and remember I said the mesos themselves are the hemangioblasts. Then you have the tube formation. That's what we refer to as formation of the blood and the blood vessels. So the first blood vessel that's formed in the extraembryonic mesoderm, lining the yolk sac, then in the connecting stalk, and in the chorionic membrane at the beginning of the 3rd week. Also, the embryonic vessel begins to form about 2 days later. Then the heart begins beating at the beginning of the 4th week. Then the cardiovascular system is the first functional system to develop. So in as much as you remember that I said that the neural tubes are not actually formed and makes these others to form, but in terms of the first functional system, the one which actually starts to function, it is the cardiovascular system. That's the first functional system to develop. Then the embryonic vessels first form mainly the aorta, and you have the gonad mesonephros area. Then in the liver, we have the definitive hematopoietic area. Then we also have the definitive hematopoietic area, which is the bone marrow. Okay, that's one of the definitive hematopoietic area. Okay, so next we are going to talk about the primitive circulation. So we have already established that the heart begins beating by the beginning of the 4th week. Then the cardiovascular system is the first functional system to develop. So we have already established this. So this, what we have established, is called the primitive circulation. Okay, so of course there are some left out materials, and when something in the book is not mentioned in the section, it could be due to some of the two reasons. It is not a significant concept to structure piece of information, or it is going to be repeated in greater detail in another chapter, or if something is not discussed in the session and we will not be included for the quizzes, but may be required at the university. So what we're just saying is there's some which we may have left out in the material, but of course these are some of the things you have to know. You don't have to you have to use a textbook, of course, to use out some of the things that may be required at the university. Okay, that's what we have said, and others may be discussed in the future sessions. Otherwise, this is our checkpoint, and if you have any further questions, you can just ask questions in the comment section. Otherwise, we are done. If you haven't subscribed, please subscribe and the bell notification, and I'll see you in the next lesson 4 to be specific.